To the Adler Museum. If you haven't been here before, this is quite a place with relics that you would have only heard about from maybe 100 years ago. And it's really interesting to imagine how medical care was back in that era. And so welcome to the third IDORI Symposium, and we're very pleased that you're here. I would just let you know that it's good that you're here in the Adler Museum, because I think this is the last time we'll actually have one of these here. Subsequent ones, in general, will be at the Marie Curie Theater, where we have more of a sort of intimate environment, tiered, terraced environment, which I think works very well. But here this is very special to be here. So for those of you who missed the first symposium for IDORI, I wanted to give you a little bit of a background. Even if you were here, this will be a good reminder. And so IDORI, Infectious Disease and Oncology Research Institute, was conceived with the idea of integrating science at Wits. So it started with an idea of integrating the vast array of infectious diseases research that goes on at Wits in different domains, different elements of science, but also geographically quite separate. And with world-class infectious disease work truly going on here at Wits, the idea was, wouldn't we be able to go further if we more integrated the work? And that led to a concept of also integrating the oncology sciences at Wits. Also, first in its class in Africa, top-notch work going on here, and also in need to be integrated. So IDORI is not about breaking silos. We don't bust silos, because for those of you who've worked on the farm, you need silos to hold the grain in place. Otherwise it just goes everywhere. But it is about interconnecting them and getting them to interact, getting what's inside to interact. And I should say actually that we are in the process of building a new campus. We purchased the Isle of Houghton, which is near here. It's right across from Rodian School and kind of across from Sunnyside. And that campus will bring together this integrated infectious disease research and also the cancer research. And the magic also happens, and what makes IDORI really unique, there's nothing like this anywhere in the world, is you take these two integrated circles and you put them together, and it's where they intersect that really the magic can happen, which is to identify novel ways to prevent, diagnose, and treat cancers, for one thing, using, studying the intersect of microbes, microbiomes, viruses, and cancer. So that gives you a sense of IDORI as a background where this symposium is occurring. But today we have Patrick Arbuthnot here, and Patrick's has done work in a variety of areas, and one area that has been a main focus that is often identified with Patrick is the relationship of, it is on hepatitis B virus, and in some ways the relationship with cancer. And just to summarize, and Patrick is going to talk today about much more than hepatitis B, but there are a lot of people living with chronic hepatitis B. WHO estimates 254 million people living with chronic disease. There are 1.2 million new infections each year. And there's a lot of deaths, between 900,000 to 1.1 million deaths. And that's principally due to chronic liver disease cirrhosis and liver cancer. And so liver cancer kills 800,000 people annually. A good proportion of those are on the African continent, and somewhere around half, you can see the range, is due to hepatitis B virus. And prevention is critical. There's a recombinant HPV vaccine, which is safe and effective. But there are other things that need to be done for therapy, and there's a lot of potential for better vaccines against hepatitis B. And so I'm saying all this because, although, again, Patrick, you may not speak heavily on hepatitis B, but it's an example of the interesting relationship of a virus like hepatitis B and cancer, and the ability we have with that knowledge to impact a scourge. Liver cancer, the case fatality rate for liver cancer here in South Africa and most of Africa approaches 90%, 95%, and largely that's because, liver cancer is one of those things that you don't want to get anywhere in the world, but here access to healthcare occurs so late that there's really very little that can be done. So the more you can do on the prevention side and the screening side, the better luck you will have from a public health standpoint. In a lot of ways, to me, that summarizes what IDORI is about when you look at that intersect. So also I just want to say that Patrick has built a remarkable lab with remarkable people. There are a number of people in this slide who have been very active with IDORI, including Kuben, I can't really point here on the far left, and Betty and Abdullah and Rebecca, and I'm sure a number of others. And so it's a very impressive lab. I think a lot of the people in that lab are going places, and hopefully those will be good places. And so with that, I would very much like to introduce Patrick. Patrick is a personal professor and director of the SAMRC Antiviral Gene Therapy Research Unit, or otherwise affectionately called AgTRU. After he completed his medical studies and a PhD, he worked on gene therapy as a postdoctoral fellow at Necker Hospital in Paris. And then on returning to South Africa, Patrick established the AGTRU, the AgTRU, which is now published widely on gene therapy-based methods to counter infection with hepatitis B virus and other pathogens. Recently, the team is engaged with WHO, the Medicines Patent Pool, various governments, philanthropies, and industry to establish an African mRNA vaccine technology platform. I think we're going to hear about today. Research in the unit is also advancing the use of recombinant adenoviral vectors and yeast express proteins as vaccines. So it's with great pleasure that I introduce Patrick Arbuthnot.
Thank you, Rob, and thank you everybody for coming to listen to this presentation. There's been a connection between gene therapy and vaccinology for several years now, and I think it was first realized that gene therapy could have uses for vaccinology when adenoviruses, recombinant adenoviruses, were first engineered during the 1990s. It was realized then that adenoviruses could function very well to deliver antigen coding sequences which then could function as vaccines. Unfortunately, the progress from the early days up until very recently was quite poor, and in fact there were some disasters because Many of you are aware of one example of a disaster, which was the Pambelia trial, which was conducted here in South Africa, which was using an adenoviral vector, adenovirus 5, to try to vaccinate against HIV. And the disaster was the fact that the vaccination caused an increased risk of contracting HIV, which was a pretty serious problem. The trial had to be cancelled as soon as this became evident. And it was only really when the pandemic struck and the adenoviruses were used again to vaccinate against SARS coronavirus 2 that they were shown to be pretty good at preventing infection with that particular serious infection that we know about. Of course, there was also the development of mRNA vaccines at the time of the pandemic, and that's come on in leaps and bounds and is a very important part of modern vaccinology, and of course it also has its background in gene therapy. So what I'm going to speak about is really an overview, summary, high level, not too technical, although we really love talking about technical things, so if you want to know more details, feel free to come and speak to me or one of the other people in our lab, and we will explain the details if you want to know about them. So that's not what I will be speaking about, it will be a high level presentation. So to start with, what is gene therapy? The term gene therapy was coined in 1972, which is obviously a long time ago, 52 years ago. And the father of gene therapy is a person by the name of Theodore Friedman, or Ted Friedman, who probably most of you don't know. But anyway, he was the first author of this paper, which interestingly had a title that was a question, Gene Therapy for Human Genetic Disease, ? And he really, in this article, speculated about what the possibilities were for using a gene as a therapeutic. And I say speculated because this was very early days. Remember, the first plasmid clone was only generated in 1973, a year later, a year after this particular paper was published. And other things like sequencing, Sanger sequencing, that was only described in 1977. And of course, PCR was way in the distant 1985. So all these very important fundamental techniques of molecular biology hadn't been established, which meant that Ted and his colleague Richard Roblin were really, as I say, speculating about what the possibilities were for gene therapy. And I went into a lot of detail about how this gene therapy could evolve, what it could be used for, but essentially they were focusing on using gene therapy to cure inherited diseases, and their interest was in a disease called Lesch-Nyhan syndrome, which is caused by a deficiency of HPRT, hyphosphorylatolyl transferase, an enzyme that's important for salvage of purines. And these children, usually boys, of course, because it's sex-linked recessive, have very high uric acid and mental retardation as well. And they were trying to cure this disease, but using very primitive methods, and only cure in cell culture, should I say. And they were doing what I think, in retrospect, is crazy, trying to take all of the DNA from cells that were normal and transfecting that into the diseased cells and seeing if they could find evidence of a cure of this Lesch-Nyhan syndrome. And they acknowledged that the methodology was flawed, and understandably, because it was such early days when they were doing this. But anyway, the term gene therapy was coined then, and it's stuck. It's become a popular field of work, as you probably all know, and the name gene therapy hasn't changed ever since that time 52 years ago. That's a picture, obviously, of Theodore Friedman, Ted Friedman. He's still around, and he's active on the editorial board. He's an editor, in fact, of the journal called Gene Therapy. But of course, his paper was prescient because he knew a lot of things which we mentioned, a lot of things that came true later on. One of the things that came out in his paper, which of course is very important, was all the ethical considerations related to gene therapy. At the time, there were people who were interested in eugenics, which is obviously not a very savory kind of discipline, really. People who are interested in trying to improve intelligence or whatever other properties of humans by using various methods such as possibly gene therapy. And this is one example of a figure which I think is very important because it just demonstrates how good he was at anticipating what was going to happen, and here you can see that there's a diagram of a virus and other types of DNA being introduced into a cell to have a curative effect on whatever disease it was, Lichtenheim or whatever. But yeah, this was published a long time ago, but it's actually still quite accurate. Now, gene therapy has obviously evolved enormously since those early days. It's hardly recognizable now compared to what it was like then. And really, some of the important features about gene therapy are that it employs rational design to develop new drugs. And the rational design essentially is based on knowledge about sequences. Obviously, sequencing is a huge field now. Next generation sequencing is very, very sophisticated, and huge amounts of data can be generated which can be used to inform the design of new therapeutics. And the other important thing about gene therapy, just to know, is that it uses nucleic acids as a therapeutic and not a small molecule or any other kind of drug. So it's important to understand those 2 points about gene therapy. The field has expanded massively, as I said before. The introduction of new methods and new techniques, discovering of new procedures has had a huge impact. For example, gene silencing in the late 1990s, was it? Was it then? Yeah, I can't remember exactly. But then gene editing, of course, and CRISPR-Cas, especially with the work of Jennifer Doudna and Emmanuel Charpentier. The people who discovered silencing, Craig Miller and Andrew Fire, who were awarded the Nobel Prizes, was Jennifer Doudna and Emmanuel Charpentier. So those were very important developments in the fields of molecular biology that had an impact on gene therapy. Of course, gene therapy makes use of artificial genes as well. mRNA, I just mentioned, has had a huge influence on gene therapy too, especially in the vaccinology field. And synthetic DNA and RNA is also quite important. And the other vital thing that has had an influence on gene therapy is the improvement in vectorology. Now, DNA and RNA, the active components of gene therapies, are highly negatively charged big molecules, and they don't get into cells. It's not like taking paracetamol or brufen, which are small molecules that diffuse into cells very readily. These nucleic acids are negatively charged, and they're repelled by the cell surface membrane, and they can't get into cells very easily. So it's important to use a carrier or a vector to get the DNA or RNA into a cell. And usually the carriers are viruses, such as the adenovirus I just mentioned before, or a non-viral vector, such as a lipid nanoparticle, which encapsulates a piece of RNA or DNA to deliver to a cell. Now, if you take another step back and think about the implications of what I'm talking about here, if you can alter the function of a gene, say for example by silencing or editing, that is a profoundly important tool to have at your disposal, because basically all living organisms are dependent on their genes. The way we are is because of what our genes do, the way we function. And it's true for all biological organisms, basically. And you can think of viruses basically as parasitic genes. They really cannot function outside of a cell. They have genes that get into a cell, and they have replication mechanisms encoded by those genes which enable the viruses to replicate. So if you can disrupt or influence that genetic function, then you have a huge tool, a very important resource to be able to treat a variety of diseases, whether they be infection or cancer, as Rob was talking about earlier. Cancer is a very important disease. Cancers are caused by genetic defects, and if one could change those defects, then obviously you have a very important tool, ability to be able to treat disease. Now, having said that, the progress with gene therapy has not been plain sailing by any means. There have been some major issues with gene therapy along the way. As I said, it quickly captured the public imagination. The use of the term gene therapy was quite catchy, and people bought into it very quickly because it was quite easy to understand what it was about. But the field was very severely criticized for overstating the success possibilities of gene therapy. And there was a study commissioned by Harold Valmus, the then chairman of or head of the NIH, and he was quite scathing, or the study was quite scathing, the report, when it said that there was a mistaken and widespread perception of success that the people working in gene therapy tried to convey to the general public, which was obviously quite a serious thing to say about the field of gene therapy. And that was in the mid-1990s. And to make matters worse, there was a clinical trial in 1999 when a patient called Jesse Gelsinger, and you can see the picture of him there, an 18-year-old, who was treated for an inherited disease. I forget what it was, but anyway, it was a mild disease, and you can see he's pretty healthy looking there, and he was able to, the ornithine transcarbamylase deficiency, he was able to control the problems related to this disease by modulating his diet. The issue was a high concentration of urea, but if you decrease the protein content that he ate of his food, then he was able to control it. But he somehow wound up on a clinical trial for the treatment of this disease, and he received an adenovirus vector in high amount into his portal vein to get into the liver to treat the disease. And he developed a cytochrome storm, and he died as a result of this. And it was a very serious negative impact on the field. And then there was another important bad event for gene therapy, which was related to the treatment of SCID children, severe combined immunodeficiency children. And these children, they were treated in Necker Hospital, which is where I did my postdoc. And these children have a deficiency of IL-2 receptor gamma, which is important for the maturation of T and B cells, and they have severe immune deficiency. And these children were treated with recombinant retroviruses, which integrated, and unfortunately, some of the integrations were at sensitive sites and caused leukemia in the kids, which was a very unfortunate thing. At the time, there was, I just came across this, I didn't realize it, but John Travolta acted in a film called The Boy in the Plastic Bubble, which was about, some of you remember it, about these children who had to live in plastic bubbles basically to prevent them from being infected by the pathogens and non-pathogens really that we come into contact with every day. So the restoration of confidence was very slow after that, because there was this report from the NIH, and then after that there were these incidents as well, which were quite serious, that had a negative impact on gene therapy. So it took a long time for it to get back on its feet. So as far as my personal involvement is concerned, how did I get involved with gene therapy? It was in the early 1990s that I finished my PhD, and I was looking around for ways to broaden my horizons, and it was recommended by my PhD supervisor, Lynn Fitchin, and also Michael Q, who most of you know is a famous hepatologist from Wits. And they recommended that I go and work in the laboratory of a person called Christian Brescio, who was working at Necker Hospital in Paris. And I went there not knowing quite what was going to be in store for me, but when I got there, this is where it is. It's, you can see that arrow there right at the bottom is just within about 5 or 10 minutes walking distance from the Montparnasse Tower, which is that sort of red rectangular symbol down at the bottom around about 7 o'clock on this picture of Paris. And I worked in this unit, INSERM unit. INSERM is the Health and Medical Research Institute of Paris, or France, should I say. It's equivalent to the MRC or the NIH in other countries. And I went there, and I didn't know what to expect, as I said, but Christian was setting up this subgroup within his unit, which was going to be aimed at developing gene therapy. And the person I worked with was Nicola Ferry, who just recently transferred from from the Pasteur Institute, and he was very good at making recombinant viral vectors, particularly retroviral vectors, and also had an interest in adenoviruses. So that's what we started working on at the time that I started. And it really struck a chord with my heart because it was so interesting and appealed to the sort of engineer in me. I've always liked to sort of tinker and work in the lab and try and manipulate things to get them to do what I want them. And I think there are a lot of people like that around who like that sort of thing. And this was really something that I enjoyed. And I definitely sort of realized quite soon that was what I wanted to do. It was what I wanted to carry on doing when I came back to South Africa. What we did was to try and target the therapy for liver cancer, again coming back to cancer, Rob, and the clinicians in the audience will know that a common feature of liver cancer is that the cells dedifferentiate and overexpress alpha-fetoprotein. So the promoter of alpha-fetoprotein in adults is switched off, but in the fetal stage, the alpha-fetoprotein is switched on. So these cancers revert to the dedifferentiated state. So we took the promoters from alpha-fetoprotein and coupled them to a potentially toxic gene encoding thymidine kinase, which would convert a harmless prodrug called ganciclovir to a toxic product that would then kill off the cancer cells. It was quite a complicated idea, and again, it was fairly early in the stages of gene therapy, but we got it to work. It worked at least in cell culture. But I think that the vectors are not ideal for use in patients. But anyway, that was my introduction, and it was a very good introduction to gene therapy, which stuck with me ever since that time. And it was a very valuable experience working in Echo Hospital. When I returned to South Africa, obviously it started off as a very small team. I had a few honor students too, I think it was. And some of you will know Sergio Carmona. He was the first honor student I had. So when I got back to South Africa, he was part of this three-person team. And from then it built up, and there have been many people who are quite talented who have stuck with the unit. And two of them, I think, deserve particular mention. Abdullah Eli and Kristen Bloom. They were two PhD graduates from the antiviral gene therapy research unit. And then two other people who joined who weren't PhD students of our team were Betty Mahatma and Ben Naidu, who I'm sure you all know these four people. Of course, back in South Africa, there was much skepticism about gene therapy. But over the years, we built many technical capabilities, and in particular I've listed theirs, I think, which are the most essential components of gene therapy: viral vectors, lipid nanoparticles, gene silencing, gene editing, making artificial genes, which is what essentially was doing in Paris, and making mRNA and also self-amplifying RNA as well. So we built up a big toolbox, I think, over the last 25, 30 years to enable us to do our work. And as Rob said before, most of our work is on hepatitis B virus. It's a very interesting virus and a very underappreciated virus. And Rob, I think you used the same source as I did. So, we've all heard these figures before, and the virus is spread throughout sub-Saharan Africa, as you can see from this heat map on the world, and also East and Southeast Asia, where the infection is particularly common. A statistic that I don't think you included in your introduction, you might have done, was that there were 1.1 million deaths. In 2022 there were that many deaths, and most of them are from cirrhosis and hepatocellular carcinoma. And that number of deaths is actually equal to or slightly higher than the number of deaths that arise from HIV complications, which people don't appreciate. And I looked at the figures for funding of HIV research versus the funding of hepatitis B virus research, and it's around about 365 times more for HIV than it is for HBV. So for one day's research in HIV, that cost is basically a whole year's research for hepatitis B virus, which is an interesting statistic, I think. The available treatment is rarely curative, that's a problem, and one of the main issues is with the replication intermediate, which comprises covalently closed circular DNA. So this is a very stable circular piece of DNA that is untouched by available therapy. So lingers in the hepatocyte and it doesn't go away. It's something like the provirus of HIV as well. So it's an important disease, and HCC, as Rob also mentioned, is a highly aggressive tumor, and people die almost always as a result of the disease. When I worked in at Chris Van Baragwanath many years ago, the average time from a patient presenting with symptoms to death was 6 weeks, which is extremely rapid. And the average time from onset of symptoms before coming to the hospital was 8 weeks. So the malignancy is an extremely aggressive one. And it's important that hepatitis B virus should be cured to prevent this risk factor for liver cancer. So we focused on developing gene silencing and gene editing to offer more effective treatment of HPV. And we worked initially on adenoviruses for HPV therapy, and this is an example of one set of data which comes from Carol, and also Carol Crowder, who's here in the audience, and also Betty Maeb, who I pointed out earlier. And this was a study using adenoviruses. These are helper-dependent adenoviruses, so we thought we'd try and get around the problem of the strong innate immunostimulation that comes with adenoviruses by making helper-dependent adenoviruses. So all the genes were stripped out of the adenovirus, so it was only really just had the therapeutic silencing cassette. And you can see that there was quite good silencing of this particular marker of hepatitis B virus, which is replication, which is the surface antigen. And if you look at the liver and you stain for the core antigen, you also get a pretty good inhibition of mark of replication. And these were in transgenic mice. It's a very stringent model of hepatitis B virus replication. And then we extended that to look at vaccination. And of course, just before I go on, the silencing, of course, has no effect on the cccDNA, which is problematic. We've used these, this is more recent data, these adenoviruses to vaccinate, and you can see there in the red line that is almost similar to the blue line. The blue line is the licensed hepatitis B virus vaccine that's available commercially and all babies get in South Africa. And the adenovirus vaccines work basically just as well as the commercially available ones. So the problem with the cccDNA could be overcome by gene editing, and that's what we focused on next. And we used artificially engineered sequences which are called TALENs, transcription activator-like effector nucleases. And they are based on a type of protein from a plant pathogen which is called Xanthomonas. These plant pathogens produce these proteins that interact with the DNA of the host plant DNA, and they activate genes which are favorable for the replication of the Xanthomonas microorganism. So we made some TALENs and we targeted them to specific sequences of HPV and obviously try to avoid endogenous human sequences. And this is basically what it looks like. I'm not going to be too technical, but you can see there those light blue rectangles in the middle. Those are little monomers that are strung together in tandem, and they identify a sequence or a particular base of DNA. So you have a sequence like this, the NLS refers to nuclear localization signal, and then the DNA binding repeat domain, each subunit binding to one base, and then there's a FOC1 nuclease. So the nuclease cuts the DNA. And it's a diamond. You need to cut both strands of DNA, so you have a left and a right TALEN, and they target sites close to each other on the DNA, and they cut, and if both of them cut together, they generate a double-stranded break. So the rationale behind using this was that by repeatedly cutting the DNA, it would be repaired by error-prone non-homologous end joining, and then there would be an error introduced into the DNA. I'm not going to speak about CRISPR-Cas, and I know that everybody thinks that gene editing equals CRISPR-Cas, but it doesn't. In our experience, these TALENs, we worked on CRISPR-Cas as well, these TALENs work considerably better than the CRISPR-Cas. I don't know if it's a property of these particular ones that we designed, maybe we got lucky with it, but they really do work a lot better than the CRISPR-Cas that we've tested. When we looked at how good these were in vivo, And you can see again, this is a hepatitis B surface antigen assay to analyze replication. And you can see the surface talon basically knocked out the expression of surface antigen almost completely. This is in a mouse model of hepatitis B virus replication. And when you use the core talon, which doesn't target the surface, you don't really get such a significant inhibition of surface antigen, which is good because it confirms that these are working according to the design. And we got confirmation using other markers as well, viral particle equivalence and core antigen, and we also verified that the target sequence mutation was occurring by looking at specific regions that were supposed to be targeted. We also found no evidence of any off-target effects, which was reassuring, but of course that's actually very difficult to prove conclusively. But the talons encoded by DNA are a little bit problematic because DNA is quite stable in a cell, and if you have the talon hanging around for a long time, so weeks or maybe months in a cell, then the chances of off-target effects are increased considerably, and that's certainly something you want to avoid. You don't want to have any genotoxicity as a result of the DNA hanging around. Of course, the DNA could also integrate. So it was around about this time we published this work, and it was picked up by Johnson & Johnson Innovation, and they approached us and said, we like your work, but we don't like the fact that you're working on DNA. We think that you should use your talons, but code for RNA using these talons, because it's more drug-like, it's shorter half-life, you can regulate the doses, and there's no genotoxicity associated with making using mRNA to treat the disease. So we obviously jumped at this opportunity, and they said they'd fund us to set this up, which we did. And yeah, that was the start of our work on mRNA. And just to carry on about all of the mRNA side of the anti-HBV work, we obviously needed to put them into non-viral vectors or lipid nanoparticles. And to test the delivery efficiency, we use a reporter gene which is called firefly luciferase. So fireflies, as you all know, are these insects that fly around and they produce light. And we can use the firefly luciferase gene, make mRNA from it, and that's a very useful, very valuable marker to test how well the mRNA is working. So this is an example of delivery of mRNA coding for firefly luciferase, which was injected intravenously into these mice, and it goes to the liver. This is work from Tiffany Smith, I think is in the audience somewhere. There she is, yeah. This is her PhD work, and that was very, very strong signal, as you can see there. A lot of light coming from the livers of these mice, which was very pleasing. And when we looked at the replication, these are the circulating viral particle equivalents, again in transgenic mice, you can see that the number of circulating viral particles is basically almost completely wiped out, which is hugely impressive. I think that this is quite a significant breakthrough for us, and it could, I think it deserves to be taken forward to be assessed for clinical trial utility. So obviously this was very useful when it came to the pandemic, and mRNA vaccination was so important, was becoming so important. So that's how we became involved in the mRNA platform. So something that the WHO picked up on and everybody in the world picked up on is that the health security in LMICs is very poor. And mRNA technology provides a powerful and easy way to overcome some of those problems, particularly in the vaccinology field. So they, the WHO Medicines Patent Pool, they established a technology transfer program, and that's what we became involved with. And the person who drove this was a person called Martin Frieda, who some of you may know. I see Rob nodding his head. And he has done this very efficiently and has been very, very supportive with driving the establishment of the mRNA platform. And the idea, of course, was to enable LMICs to make their own drugs so that they wouldn't, or own vaccines so that they wouldn't have to rely, I mean, it's an old story, everybody knows this now, that they wouldn't have to rely on the global north. And I just came across this article, this was fairly recent, and it just reinforces this point because sometimes I think people forget about about it, but I think it's worthwhile just reminding everybody. This was in Business Day on the 22nd of May this year, so it's just under 2 months ago. And you can see that the headline there is "SA Bullied by Makers of COVID Vaccines." And you'll have to go and read the article if you're interested. But really what it summarizes is how South African government was on the back foot when it came to negotiating. It was a very difficult position for them to be in because the population needed to be protected against SARS coronavirus 2, but it didn't have the means to implement its own vaccination program. So Moderna, for example, was demanding $40 a shot of its vaccine, which was a heck of a lot of money. I think Pfizer-BioNTech asked for $10, so obviously I went for Pfizer-BioNTech, which most of us have received. So that was something that came out recently, but it's worthwhile making a point about that. So we were asked to provide the technology support to make a COVID vaccine. And the starting point was to make a Moderna-like vaccine. And the attitude of the MPP and WHO was that investment in science and community involvement are crucial, and they kept on hammering that point, and also making sure that we understood that the generous support they provided was not for the long term. We had to make a sustainable case, or case for sustainability ourselves. So the first vaccine we made was, as I said, a SARS coronavirus 2, which we used public information, publicly available information to design. And this is just an example of the titers of antibodies against the SARS coronavirus 2 spike protein. So the vaccine worked. So that was a huge relief. And I think we did pretty well, because from the start of wanting to make this vaccine to concluding this preclinical study in mice took us 6 months. So for a starting point, it wasn't too bad. And I think we'd do it quicker if we had to try again. But of course, the big time-consuming part is the assessment of immunogenicity in the animals. We also tested the neutralization, and please don't throw up your hands and say you can't see what this is all about when you look at the next part, which is this part. I always tell students never to do this, but anyway, I've done it myself. But don't focus on the content of this table, just look at the colors. And the color red indicates a very strong immune response, a neutralizing effect. And you can see that some of the vaccines, which are sort of burgundy or maroon, have a very strong neutralizing effect on the pseudovirus carrying the spike protein. So this was all very encouraging, and it was a success, I believe. And this is passed on to Afrigen, the Cape Town firm, to take it further into clinical trial. We then obviously looked around to work on other vaccines using mRNA, and a project that we've been very involved in, which is a very big project, was making mRNA vaccines against TB. And this was done in collaboration with a team from University of Cape Town, Tom Scriba and Munya Musfosfi. And they did a study preceding this project, which was based on the notion that there's an immunological advantage in controllers of MTB infection. And they tried to work out what this advantage was in their study. And they did a very detailed analysis in partnership with a group from Stanford, and they did single-cell sequencing of T-cell receptors in progressors and non-progressors. So progressors were people who developed active TB, and controllers who didn't develop active TB. So both groups were infected with TB, but the controllers didn't get it. So they did this next-generation sequencing, single cells, etc., and their analysis enabled them to identify certain T-cell receptors that were common in controllers, and they could then identify the cognates of those T-cell receptors, and the cognates being antigens from MTB itself. And obviously those should be good vaccine candidates. And of course, it's important to understand, I think everybody knows that CD4 T cells are vital for control of MTB infection. So they looked at the CD4 cells for these T cell receptor sequences. So they identified 4 important sequences, which are called PE13, PPE18, CFP10, and WBBL1. So these 4 antigens were the ones that we thought, or they thought, were the good ones for mRNA vaccines. So we designed mRNA vaccines
encode these particular proteins. We decided at a fairly early stage that we didn't want to be encumbered by treading on other people's intellectual property, so we developed our own proprietary backbone, and Abdullah is the one who really is credited with designing that. He's a real dab hand when it comes to applying molecular biology, and he made this proprietary backbone plasmid, which we've used extensively now, and it works perfectly every time. So we made vaccines or mRNA templates that would encode individual mRNA sequences, and this is a beautiful gel, capillary gel electrophoresis, which is done by a postdoc in Jabula, Nyandu, sitting over there with a stripy shirt. And you can see these 4 antigens encoded, the mRNA encoded by these, encoding these antigens work very nicely. You can see lovely thin pencil lines, which indicates that this is pure and intact messenger RNA. And when we looked at the function, this is the mRNA for CFP10, and we did immunofluorescence analysis, and you can see that the CFP10 protein lights up very nicely when you introduce the CFP10 mRNA into those cells, so it was working well. We also made polyprotein constructs as well to make a simpler version, or multimeric version. And we tested these in mice, so-called Kramnik line, and we looked at the secretion of cytokines in response to stimulation with various peptide pools, and I'm not going to go through this in too much detail because it gets a little bit too much detail. So essentially what this boils down to is that the stimulation of the appropriate peptide pool led to the secretion of a cytokine, which it should have done. And this is just a tabular heat map type thing to represent those data. And interestingly, this strain of mice, which is called Kramnik, which I hadn't worked with before, is a very good model for simulating HPV infection. So they work better than the Black 6 and the Bulb C, and we've now got them in our animal facility. So you can see the Kramnik are the best model. And these vaccines are also protective against challenge. So Kramnik mice, this was done in BSL-3 level safety in UCT. These animals were vaccinated and then challenged with TB microorganisms. And the vaccines, the mRNA vaccines, actually provided protection against the challenge with the MTB. So we've expanded our range of mRNA vaccines now, and we're also working with Glenda Gray and Linda Gale Becker with a brilliant consortium to make HIV mRNA vaccines, and that's another exciting project which I'm not going to go into detail. It's at an earlier stage now, but it's getting going quite nicely, and it's quite promising. The last topic I want to deal with is something that's I find very interesting. This is a really nice project that we have embarked on in collaboration with a group of synthetic organic chemists on main campus. And they had the idea of trying to make lipid nanoparticles that are green, that are environmentally friendly, and also affordable for use in low and middle income countries, and also ones that could be formulated quite easily. And something that's not fully appreciated is that the ionizable lipid, which is a very important component of lipid nanoparticles, such as found in the Moderna vaccine or the vaccine that we would have received from Pfizer-BioNTech, those ionizable lipids are derived from products of the petroleum industry, which is obviously not environmentally friendly. And if you're going to continue with making mRNA and delivering it, it's going to be a major treatment for people, then it's not good that we should be using petroleum industry products for that purpose. So they've been working on a different approach, a green approach, and we also, in our discussions with them, we wanted to make sure that we would be able to make these ionizable lipids so that they're free of IP restrictions, say from Moderna, Pfizer-BioNTech, or Chirus, or all these other companies that are making ionizable lipids. We wanted to make them stable, they needed to be cheap to make, and also easily formulated. So what Charles de Koning is the person who is leading the team from synthetic organic chemists. He's been working on cashew nutshell liquid, which is very interesting. This is what a cashew nut fruit or nut looks like. So the red orange part is called the apple of this. And then hanging underneath the apple
is a shell that's encases the cashew nut that you can buy from the supermarket. And that shell is interesting because it contains oils. And those oils are very useful for reasons that I'll explain in a moment. So there's an abundant supply of cashew nut shells. About 450,000 metric tons of cashew nuts are produced per annum in the world. And it's an agricultural waste product, so these shells are really thrown away largely, and sometimes they burnt as fuel. But it's widely available in Africa and other LICs. And if you look at this pie chart here to indicate the distribution of people, countries producing, or regions that produce cashew nut shells, you can see that West Africa particularly produces a lot, and East Africa also produces a fair amount. And collectively, Africa produces 54% of the world's cashew nut shells, which is quite an important thing to know if you want to use the cashew nut shell well. We don't have cashew nut shell crops here. I think there's some in Mozambique, but it's mostly north of us on the east and west coasts. The useful thing about cashew nut shell liquid is that it's only got 3 lipids in it: anacardic acid, cardinal, and cardol. And this is unlike other oils such as peanut oil or olive oil, which contain a whole lot of different lipids, which make it difficult to purify a single lipid to make the product that you want to make from it. And the other useful thing is that it's abundant. The amount of cardinol, which is the ideal product that you get from these, ideal precursor that you get from these shells, is about 0.5 to 0.8 grams from one shell. That's quite a lot if you consider how much there is that's going to waste. So if you just to illustrate exactly how this process works, so this is an example I think of the streets of a town in Tanzania, where you can see all these cashew nut shells lying on the street. And you can fairly easily extract the oil from these shells, just to try and require sort of crushing and filtering, and then there's a distillation process as well. This is what the oil looks like. And then the process entails hydrogenation, which just entails treatment with hydrogen peroxide to generate a hydrogenated cardinol. That's the pure product that you can use to make the ionizable lipid that we're so interested in. So from that hydrogenated cardinol, the synthetic organic chemists go through various steps to make different ionizable lipids, and there are all sorts of different variables that they introduce into these lipids. Now we've got a library of about 50 of these ionizable lipids, and they've got different properties such as different ionizable groups, pyrazine and other things as well. Varied chain lengths, they have saturated, unsaturated chains, fatty acids with and without ester bonds, and there are a whole lot of other different things as well which I looked into. Others, so we've got a library of about 50. And we can use these to make lipid nanoparticles carrying mRNA. And the classic method is using microfluidics. And Dylan, who's also in the audience, is the person who's responsible for this, and he does it very well, where you mix a whole lot of different components, the lipids, cholesterol, pegylated lipid, with the RNA, and you mix it at high velocity in this microfluidic system, and then you get a lipid nanoparticle at the end of it that you can use to inject into mice. But what we've also been working on, and this shows some promise, it's not as good as the 4 components of the microfluidic method, is to just use one component, one ionizable lipid plus the RNA, mix them together in a simple formulation using a vortex mixer, which is obviously ideal if I can get it to work, because that would be suitable for an African context. We don't have to spend a huge amount of money on these microfluidic reagents. So when we look at these particles that are produced, you can see that you get a nice size. It's around about 90 nanometers in diameter, which is good. And you can see that the bell curve is quite tight, which means that the so-called polydispersity index is good. So they have a uniform size and there's not a big spread or multiple peaks that could be problematic. And we also found that the encapsulation efficiency, the amount of RNA that gets into the lipid nanoparticle, is also very high. You can see here it's around about 90%. Dylan sets very high standards, and he says it must be 90% or higher. Doesn't accept anything lower, but most people do. So these still work very well as encapsulators. That's obviously quite important because you've got to have the RNA lying outside of the lipid nanoparticle because it'll just get degraded and it also will affect the dose. So when we tested delivery in vivo, we tested firstly the Moderna lipid. This is a lipid formulation containing SM102, which is the Moderna ionizable lipid, and you can see it delivers very well. But an important thing is that it gets into the liver as well. That's, I think, something that's undesirable. We want to have the formulation going just to the site of injection. When we tested our lipids, this is an example of one of the 50 that have been generated, you can see that when it's injected intramuscularly on the left, in the left hind limb, and then intradermal on the right, those two mice where you can see on the flank of the animal, the expression of the reported gene FLUC is localized to the liver, which I think is a very important property, a very good feature. So we next tested the ability of these formulations to induce an immune response, and we did the same kind of study as we did with the TB study, where we looked at the cytokines released from the splenocytes to evaluate how strongly the immune response was. And you can see from this that when comparing the cashew nut derived lipid, which is indicated there in the red rectangle, you can see that the response to the release of these different cytokines, interferon gamma, TNF-alpha, and IL-2, is good and the same as the Moderna type formulation, which is just on the right. So the values for the release of cytokines in response to these peptide pool stimulations was equivalent with the mRNA or the Moderna formulation, which was very, very encouraging. And you can see that in the controls, which are, this is a negative control, you can see that there's no induction of an immune response with those peptides. Those peptides don't induce a response, and the positive control is also good for both the Moderna and the cashew nut shell-derived formulation. So the antibody titers and neutralization data are something that you're probably wondering in your mind. Those are in the process of being analyzed and generated. And I think that it's probably not going to be ideal straight off, but certainly the results so far are very encouraging. And I think that this is potentially something quite big. If we could make something that was economical, easy to make, and by the way, the cost Dylan worked out with the synthetic organic chemist is about a 15th of the cost of SM102. So the price for one of the lead cashew nut lipids is a 15.15 of the Moderna SM102. So then, just to conclude, I think it's true to say that gene therapy is now reaching the mainstream of modern medicine. And when I was thinking about the title that I gave for this talk, and I'm just wondering whether convergence is really the appropriate term, and whether gene therapy itself is the appropriate term, because I think that what is becoming important is that ability to deliver DNA and RNA to cells is becoming very, very important, and that is what has got application in a wide range of fields. It could be vaccinology, it could be oncology, it could be all sorts of different kinds of treatment that are applicable using delivery of nucleic acids. The field is, as I've just said many times before, that it's moving very quickly. And one of the interesting new developments is that machine learning is becoming more and more prominent. I recently attended the ASGCT meeting, American Society of Gene and Cell Therapy, and that was very clear there that people are using AI to inform how to make new gene therapies, and I think that's something that we need to look out for and keep abreast of as well. There are new frontiers that are continuously being met, especially for cancer therapy, and I mention this again because Rob was particularly interested in the interface between cancer and infectious diseases, and I think that IDORI has an important role there. One of the big fields of cancer therapy that is huge, certainly at the ACCT, is the manufacture of CAR T cells. They are very, very good and potentially can cure cancers, and the advances are hugely impressive. And I always say this, and I think it's true, and I hope that you will agree with me, that it's essential for LMICs to be involved in this kind of work. You can't just wait and sit back and let global North, Europe, North America, parts of Asia come up with the treatments that we need, or vaccines that we need for the people in Africa. Now, obviously, these are new things. People are a bit suspicious. Maybe some of you in the audience are suspicious, and I think that it's quite important that there's community engagement to make sure that people are engaged fully and understand what the new technologies are. I've been very impressed in our involvement with the brilliant consortium, that's Glenn Graham and Gail Becker's project with mRNA vaccines for HIV, because they have got a group of people who are sociologists who go into the rural areas and they are constantly engaging with people to educate them about what new technologies mean. So they have radio advertisements explaining things, and I think that's highly commendable. I think that's the way to go, because you can't just expect communities who perhaps don't have such good education to immediately accept that mRNA vaccines are the be-all and end-all, because they will just won't accept it. And I think that's crucially important to make sure that that happens. And then lastly, I hope that you've gathered that this is a field that is very, very exciting. It's hugely rewarding, and it's a nice field to be in. And I think lots of young scientists, I certainly see that every day in the lab, are enjoying the engagement with this modern molecular biology and plant discipline. And of course, there are also commercialization opportunities, which are also, I think, important for sustainability. By the way, the cashew nut story and the TB vaccines have been patented. So lastly, I need to acknowledge this is a team. This is an updated version of what you showed, Rob. I mentioned the people who have contributed, so I won't go through them again. These are the synthetic organic chemists, Charles, Robin, Sons, and Anissa. Who's missing from this photograph is Connor Stockley, who's another synthetic organic chemist who's made some valuable contributions to us. And then the TB people from UCT, Tom Screamer and William Oskowski. Obviously that collaboration is very, very important, and it's exciting because we're fast-tracking, as I think I said, to clinical trial. And then these are the funders who've really been very generous with their support to enable us to do this work. Thank you for your attention.
It was great. Thank you. Patrick, that was a fantastic talk, and I'd like to ask if anyone has any questions. And as you're thinking about that, as you said at the end, it's essential for Africa to be involved in these cutting-edge advances, and it's so nice to see really the work that's going on in your lab that is on the cutting edge. When you talked about just going back to the TB vaccine work, you worked on the encoding for these 4 proteins, and it looks very promising that there may be a vaccine that could, at least in the animal model, it looks like it will work. Can you walk us through, and you mentioned commercialization at the end, your last slide. Can you walk us through what will happen now? How to take that to kind of a clinical level? Is that in the works?
Very much so, Rob. What we need to do is to make the mRNA under GMP conditions. We need to make sure that this meets the RNA that we generate for testing in humans meets the requirements of GMP. And then, unfortunately, that is not yet up and running in South Africa yet. The Afrigen team are working very hard to do it, but it's unfortunately been a bit slow. So that's what we need to do from the point of view of taking it to the clinic. And we're obviously engaging with regulators, making sure that we know exactly what they want so that we take this forward. quickly. And obviously there's not just the mRNA, but there's also the lipid nanoparticle part of it. So we won't be using the cashew nut shell liquid lipids just yet, because that's still in a developmental phase. But that will also have to be taken to a GMP quality production.
Any questions, comments? I mean, yes. That's okay. You may have the microphone.
Thanks very much, Patrick, for taking us through the journey of gene therapy and your work, which is extremely exciting. I was particularly fascinated by your comment when you came back from France. You got a sort of a cold or lukewarm reception on gene therapy. What I wanted to know is, has that changed since you started to do this work? And certainly, I mean, for some of us who are doing gene therapy for inherited bleeding disorders, we haven't seen those barriers that you faced at the time. But I just wondered if it was specifically for infectious diseases, or were the barriers removed completely, in your opinion?
Johnny, I suppose my experience is based on anecdotes. I think, interacting with one person here, another person there, who surprisingly comes up with a very negative comment about gene therapy. And I think it's usually based on lack of understanding. People seem to think that this is some kind of eugenics or whatever that is very dangerous and could cause problems. Maybe I've just been a little bit too sensitive to that. But yeah, I still do get surprised by people who come up with very anti kind of comments who I would thought were more informed. But I mean, that's just human nature. I think there always will be people like that. And I think, it's important to take their opinions seriously and address them. And, it relates to what I was speaking about at the end with the community engagement, which I think is crucial. And it's important to educate people and make sure that the promise is there to be understood properly. But also, and I said this in my talk as well, that the gene therapy has certainly not got a flawless track record. I mean, there have been some terrible things. The Jesse Gelsinger death was very, very bad. If you look into the details, you'll see that there are many things that should not have been allowed to go through, and they were. And I think there are probably some other examples of other gene therapy clinical trials. So it's, gene therapy is not blameless in this either. Nigel?
So though you've done capsules, I mean, for gene for some of the gene therapy, you need targeted delivery of particular genes to tissues. So is there any way that those nanocapsules could be manipulated in a way so they could be targeted to specific tissue types? Yes, we're not working on that at the moment. As you saw from the data, with the cashew nut shell lipids, the delivery is localized, and we don't want anything else apart from that. But there certainly are ways. The acylglycoprotein receptor, for example, which is present on the liver, can be targeted by galactose added to a lipid nanoparticle. That's a commonly used method. And there are other methods as well. People using mannose as well to target dendritic cells, and there are various other carbohydrates that can be added onto lipid nanoparticles to target to various cells. We haven't worked on that yet, but we could well do that in the future. But it is a good question, and a lot of people do work on that.
Any other questions? Yes.
Left-sided question. So, I mean, the question is with regards to telomere shortening. And, I mean, with the manipulation of gene therapy, I mean, have we considered insertion of redundant DNA into maintaining telomere sizes? Has that been ever looked at? I mean, because it's redundant DNA material that's actually causing the problem of aging.
That's not really in my field, but it's an interesting idea. So, inserting DNA into the telomeres to prevent them from getting too short and associated with aging. I must look into that. I'm sorry, I don't know. Yeah.
Patrick, you mentioned that your TALEN-driven gene editing in that particular experiment worked better than CRISPR. What's going on there? And why do we not hear so much about that method? Could it be used for other pathogens, with working on other gene editing projects?
I think what it boils down to is that it's so much easier to make CRISPR-Cas systems that target a particular gene. I mean, you just need to make a guide sequence, really, or a template that makes a guide sequence, which is really very easy. But those TALENs required piecing together a whole lot of different sequences to make that big protein that would target a particular gene. In our case, the hepatitis B virus gene, and that was Christy Bloom's PhD topic. I mentioned Christy earlier in the presentation. So she pieced together all those pieces of DNA, and that took a long time, and it took a long time to characterize them as well. Whereas with CRISPR-Cas, you can do it in a month. I think a lot of it is to do with ease, yeah.
Any other questions, thoughts bubbling up? Don't hesitate. You'll regret it if you don't ask the question. Well, Patrick, I want to thank you very much again for such a wonderful presentation, and you've been quite busy over these 30 years or so, and it's very impressive what you're doing, and what's in a lot of ways maybe even more impressive is what I think lies ahead. It's very exciting. No one really asked about the cashew story, but I think a lot's going to happen there also over the next, what would you say, year or two?
Yeah, I think so.
Yeah. So thank you. And so we have food and drink right around the corner. You can't miss it as you're heading out. Please stay with us for a little bit so we can interact. One of the points of IDORI is to bring scientists like you together so that we interact in ways that might not happen otherwise. And you can just sort of imagine the innovation that will bubble up, especially as we get going at full stride. So thank you all for coming. Thank you again, Patrick. That's great.
Thank you.
Amazing what you're